Hello, Nutrition 115 students, and welcome to Chapter 26, or Unit 26, Nutrient-Gene Interactions in Health and Disease. Now, I just want to let you know that your book has these incredible charts regarding some definitions regarding to the field of genetics. Now, before I talk about genetics, this is a nutrition course, so I want to talk about a subfield that is found under the field of or study of nutrition, and that is nutrigenomics. Nutrigenomics is basically, and just in general terms, is the field of study in which we about how nutrients interact with genes. And as we're going to learn in this chapter, this interaction of nutrients and genes also can have an effect on general health, optimal health, and diseases and conditions. I also want you to know that there is something represented by the mapping of the human genome. So we're going to also talk about a genome, which according to this table, it's the combined term for genes and chromosomes. It represents all the genes and the DNA contained in an organism, which is ourselves in this case. It is estimated that the human genome consists of approximately 19,000 genes, okay? And also the DNA that makes up each gene has 4 different chemical building blocks, and these are called bases, and they're abbreviated as A, T, C, and G. And then altogether, our genes contain about 3 billion bases attached to chromosomes within the nucleus of cells. And according to this slide, chromosomes are structures in the nuclei or the nucleus of the cells that contain genes. And humans have about 23 pairs of chromosomes shown on the left, and then half of each pair comes from the mother and half from the father. This is also part of the same table from your book, from the first slide. So again, I do want you to know what chromosomes are for the test, what a genome is identified as, and also genes. Okay, and genomics. So genes are the basic units of hereditary that occupy specific places on chromosomes. Genes consist of large DNA molecules. I guess DNA are proteins, each of which contains the code for the manufacture of a specific enzyme or other protein. As you see here, genomics is the study of the functions and interactions of all genes in the genome. Genomics includes the scientific study of complex diseases such as heart disease, asthma, diabetes, and cancer. These diseases are typically caused by a combination of genetic and environmental factors, or epigenetic factors, we call them. Okay. And then just to get an idea of the size of the human genome present in each of our cells, consider the following analogy. If the DNA sequence of the human genome were compiled in books, the equivalent of 200 volumes, the size of a Manhattan telephone book at 1,000 pages each, would be needed to hold it all. Now, as humans, we are united as a species because we all share 99.9% of the same DNA. DNA stands deoxyribonucleic acid, and we are in part unique as individuals due to that 0.1% difference. And again, as stated here, the human genome is estimated to contain about 19,000 genes. And again, not all segments, though, of the DNA code for protein formation. Again, we have that 0.1%, which lets us have that individual uniqueness in genetic makeup, which is located in non-coding segments of the DNA. These segments, I want you to know, contain instructions for the regulation of gene activity, such as when a specific gene should turn off or on, or when a gene should increase in activity or be silenced or dormant, so we call it. These segments, again, contain instructions to turn them off or on, like light switches. They're activated or dormant. These segments vary in composition based on environmental factors such as nutrient availability and exposure to infectious agents or environmental toxins. And I'll give you an example, but my mother has battled lung cancer twice. She's had 2 major surgeries in her life that removed some cancer from her lungs. And she has never in her life smoked a cigarette or ever lived with people that smoked. But she did work a lot in environments in the agricultural fields where they used a lot of pesticides and chemicals that were harmful to the human respiratory tract. So again, she contracted lung cancer based on her environmental working conditions. Okay. Genetic traits and susceptibility to disease. So the genes individuals inherit at birth from their mother and father do affect disease susceptibility. Some genes can provide protection. Some can be good against certain diseases related to lifestyle factors, but some can actually increase your risk among people with healthy lifestyles. Okay, and even in addition to that, our genetic sequencing also accounts for like what race we are, and certain race groups and ethnic groups are also more susceptible to certain diseases. For example, African Americans are more susceptible to sickle cell disease, whereas we're now finding that people in the Hispanic race are more susceptible to diabetes, and the Native Americans can also be susceptible to more like diabetes and high blood pressure. Okay. Increased intake of omega-3 acids. So this is a very short lecture. We're going to talk about how nutrients interact with genes. So I want you to know that some examples is we're finding that increased intake of omega-3 fatty acids from fish combined with the reduction of omega-6 fatty acids from oils may decrease the inflammatory gene expression. So in essence, we're decreasing inflammation in the body that can make us susceptible to diseases. Some gene variants influence the development of obesity through subtle effects on appetite, food intake, and food preferences. Also, some of us, depending on, let's talk about macronutrients here for a little bit. So it goes with this nutrient gene interactions. So some of us are very sensitive to carbohydrates and tend to gain weight a little bit more faster. Some of us gain weight a lot. We're more sensitive to maybe lipids or fats, whereas other people don't gain weight as much with those macronutrients. So that all has to do with maybe how your genes are sequenced and how they interact with nutrients. Okay. Among individuals with particular gene types, high-fat diets we found tend to increase insulin resistance and fat stores in the liver, and increase also your risk of type 2 diabetes and obesity. And this is just regarding diet, okay? And then adults with specific gene variants who frequently consume fried foods versus adults who consume fried foods less are associated with the higher body mass indices. I also want to let you know that we're also finding a lot of correlations with genes and diseases. For example, if there's been breast cancer in your maternal line, which is your mother's line, of course, if you're a female, your risk of breast cancer is higher just because of that. But if you're a male and there's breast cancer in your maternal line, they're now showing that there's a correlation with females in your lineage that have had breast cancer and you carrying the prostate cancer gene in males. Okay, so we're finding correlations also as far as genes, not just with certain sexes. Okay, so it's not just woman now, but sometimes if a woman or the maternal line has certain diseases, it can affect the paternal line as well. Okay, or the father or the males in the genetic tree. Gene variants related to impaired folate. Let me give you some examples. So gene variants related to impaired folate intake and choline utilization can increase the need for folate and choline. So what it's saying here is certain genes are not very efficient at utilizing folate and choline. There's some decrease in function in those genes. So that can increase the need for folate and choline from the diet, and you might need more than a regular person. And then the risk of developing breast cancer appears to be lower in men with certain gene variants if they regularly consume cruciferous vegetables. Let's talk about these cruciferous vegetables. These cruciferous vegetables include vegetables such as broccoli, Brussels sprouts, cauliflower, kale, just to name a few. And they're called cruciferous because they have this crisscross-like pattern in their leaves when they're growing before they harvest, so they're called cruciferous because it's kind of like a crisscross, like a crucifix kind of pattern, and they have really strong phytochemicals that have been known to not just decrease breast cancer risk, in general just cancer, but the research is more heavy on breast cancer, but just in general cancer, even prostate cancer. So in general in cancer, and also reoccurrence. So if you've had breast cancer and you survived it, they can also decrease reoccurrence as well. And we're finding here a lot of the research used to be on women and breast cancer and these cruciferous vegetables, but now we're finding that they're also having great positive effects on men as well with certain gene variants. Genetic variation influences health and disease development. In addition, lifestyle factors such as diet and exercise also play important roles in the prevention and development of obesity, diabetes, and other common disorders. Now, before I talk about this, I also want to let you know that there's a lot of things you can do to decrease your risk of obesity, diabetes, and other disorders that are found hereditary in your lineage. So it's not always a doom kind of thing. You can do a lot with your lifestyle to keep those genes turned off. But you might have to work a little harder than someone that doesn't have that in their genetic tree. And we usually say we can keep it dormant and inactive by lifestyle factors such as not smoking, living in healthy environments that don't have harmful contaminants, eating healthy water, healthy food that has low pathogenic material, and also exercise is a very important role. Exercise is even thought to improve your genes by people that are more aerobically active. They produce more lipases, certain lipases in the cells that help digest and utilize fat a lot better. Single gene defects. So just a brief little lecture on this. There are thousands of rare diseases related to a defect in a single gene when it was being sequenced. So when something was being synthesized in your body, like let's say like a certain cell of a type, something happened in the sequencing. So we call that a defect. Many of these affect nutrient needs. Such defects can alter also the absorption of or the utilization of nutrients such as amino acids, iron, zinc, and the vitamins such as B12 and B6 or folate. If you ever heard of phenylketonuria or PKU, we might have gone through that during the sweeteners chapter and the carb chapter when we talked about sweeteners. Babies, infants, newborns are tested for PKU or phenylketonuria. They're also tested for galactosemia and other diseases as well when they're born. And they're, again, genetic diseases that they inherit genetically. So there are a lot of these tests are conducted by clinical professionals when your baby's born. And for examples of what characterize single gene defects are what I just mentioned: phenylketonuria, galactosemia, hemochromatosis, and something called cystosterolemia. And phenylketonuria, I just want to go over that one, is when someone cannot process the amino acid called phenylalanine. So you will often, if you chew sugar-free gum, or if you ingest a food that has the artificial sweetener aspartame, which the brand name is the Equal little blue packet, it contains phenylalanine. So there'll be a warning that if you are a phenylketonuric, you should not be ingesting this product. Okay. And then due to its potentially severe effects on mental development, if not treated with a low phenylalanine diet starting after birth, all babies born in the United States and many other countries, again, are tested for PKU after birth. So again, if one doesn't know that this child contains phenylketonuria or PKU, it can have severe effects on mental development, okay, if not treated early. And here are some in your books. I named the 4 disorders, single gene disorders that affect nutrient needs or utilization. I would read them briefly, but I would especially hone in on PKU because I talked about that more. But just know in general a little bit about their effects and functions and what's going on. Just talk about taste and end with this. So here's a little kitty and a dog. So I want you to know that food preferences are partly influenced by genetic traits related to test sensitivity. And then genetically influenced food preferences apply also to dogs and cats, as you see here, and other animals, and as well as humans. There are also more than 80 genes that help people taste bitter foods. For example, in some people get the set of genes that make them highly sensitive to bitter tasting foods. And then people born with a high sensitivity to bitter taste tend to dislike also cooked cabbage, collards, greens, spinach, Brussels sprouts, and other vegetables that taste bitter to them. And then people who tend to like these vegetables generally don't perceive them to be bitter tasting, okay? And the genetic tendency to reject these vegetables is likely to limit intake and therefore may be linked to diseases associated with low vegetable intake that you guys are learning in this class. So as far as doggy and cats, dogs and many other animals have a taste for sweets, but cats don't. In case you have a kitty, they don't. Somewhere during the genetic evolution, the gene in cats that enable them to recognize sweetness was actually turned off. So if you have a kitty in here, it's good for you to know that. And then lastly, nutrition tomorrow. I want you to know there's meaningful breakthroughs in the future that will be in the area of disease prevention and treatment through nutritional changes. And then the knowledge of genetic traits will increasingly be used to identify individual nutrient needs and responsiveness to dietary and other changes. And also lastly, personalized modifications of dietary intake based on genotypes will become standard practice in the clinical dietetics and medicine.